My name is Josie Bergen, and today I'm going to be talking about propagule pressure and how it relates to invasive species management. So we should start by defining what a propagule is. A propagule is a set of organisms originating from a donor region, released into a non-native environment known as the recipient region. Propagule pressure describes a propagule's likelihood of establishing in a non-native environment. It arises from the combination of propagule size and propagule number. Propagule size is simply the number of organisms released, whereas propagule number is the number of independent release events. It's simple to understand that a propagule of a greater size will have better establishment success. But propagule number is beneficial because more release events means a greater chance of the propagule finding a suitable environment. In combination, these factors create greater propagule pressure that increases the overall likelihood of establishment. It's important to note that propagule pressure is affected by many other factors. Environmental factors in the recipient region are very important because establishment hinges on the compatibility of the invasive species with the non-native environment. You also have to consider the en route conditions faced by the propagule, which might hinder its ability to establish once it arrives in the recipient region. Unsuitable conditions or the spread of diseases in transit will alter the propagule pressure upon arrival. It's important to note allele effects, which arise from the reduction of the per capita population growth rate at low population density. This means that a propagule of smaller size will have a much lower chance of establishment due to limited access to mates and reduced genetic diversity. Finally, propagule composition, or the characteristics of the individuals in the propagule, is a very important factor. For example, a propagule made up of aging, diseased organisms is going to have a more difficult time establishing, even if the propagule pressure is high. Factors of propagule composition include things like health, sex ratio, age structure, and phenotype, which all play into how successful a propagule is at establishing. So our first case study relates to the importance of propagule composition on establishment success. Research done by Geramo and Bengston regarding the importance of age structure in life history found that invasion probability is highly variable between type 1, 2, and 3 organisms based on their age at introduction. The main findings from this research were that type 3 organisms, which includes many fish, insects, marine invertebrates, and plants that have high mortality, were shown to be more sensitive to age variation in propagules than type 1 organisms like large mammals. For type 3 organisms, which face high mortality in their juvenile stages, the optimal age for introduction was older than for type 1 organisms. For type 1 organisms, the optimal age at introduction was more consistent with the age at first reproduction. Also, there was important interactions between transport pathways and propagule age structure. For example, the brown goby is transported via ballast water, predominantly in its larval stage. That's because it's the only stage at which the brown goby is small enough and present in the pelagic zone to be picked up by the ships. This case study shows that there are many factors that interact with propagule pressure to affect the establishment success of a propagule. One tool used in invasive species management to assess propagule pressure is the dose-response curve. In this model, dose describes propagule pressure and response describes the probability of establishment. The linear line represents a scenario in which there's continued benefit to releasing or prohibiting the release of every separate individual. The dose-response curve, on the other hand, contains a threshold for marginal benefit, after which point management efforts to prohibit or promote establishment will be unsuccessful because there's no greater probability of establishment. So the dose-response curve serves as a tool to inform management of invasive species according to their propagule pressure. Biocontrol is of particular interest to the topic of propagule pressure. We define biocontrol as the intentional release of non-native species into an environment to serve as a natural enemy to an unwanted pest. Much of our current evidence for the importance of propagule pressure to establishment success comes from cases of intentional releases of organisms as forms of biocontrol, given that we have much more data on this than unintentional releases. Biocontrol continues to be a controversial subject due to historical instances in which biocontrol agents became invasive species themselves. Think of cane toads or mongoose. However, our continued understanding of propagule pressure has allowed for successful implementation of this management strategy in some cases. One such example of successful biocontrol is the Vidalia beetle. Rhodolia cardinalis, known commonly as the Vidalia beetle, was introduced from Australia to the Galapagos archipelago between 2002 and in 2005 as a means of biocontrol against the invasive Icera purchasi or cottony cushion scale. Cottony cushion scale is an invasive plant species native to Australia that was first identified in the Galapagos in 1982. It was responsible for extensive damage to endemic flora and fauna, namely causing disfigurement to honeydew. So, the Vidalia beetle was introduced to dampen cottony cushion scale populations. The Vidalia beetle was a suitable choice because it was unlikely to prey on non-target species, as they were unsuitable food sources. The Vidalia beetle was also favorable because the cottony cushion scale serves as its site for feeding and for reproduction, meaning that the beetle was putting constant pressure on the cottony cushion scale throughout its lifetime. This resulted in a substantial suppression of the cottony cushion scale across the Galapagos. A post-release field evaluation by Hoddle et al. Found approximately 60 to 98% reduction in the cottony cushion scale, dependent on host plant and habitat. There was also no significant non-target effects resulting from the introduction of the Vidalia beetle. Therefore, the Vidalia beetle did serve as an effective form of biocontrol in this instance. In conclusion, propagule pressure is the result of propagule size and number. Propagule pressure is affected by factors like propagule composition, ecological compatibility, and allele effects, which alter the establishment success of a propagule. Propagule pressure informs management of invasive species by helping us understand the potential impact of a non-native species on a native environment. Tools like the dose response curve help us visualize a propagule's probability of establishment and the thresholds for growth that a propagule has. This is important in the field of biocontrol, where our knowledge of propagule pressure is used to strategically act on a population of an invasive species to produce a positive outcome.